Thanks to everyone for joining our next featured panel. I'm Gbola Amusa, partner and a biotech analyst at Chardon. As discussed in yesterday's panel, The Future of Manufacturing, the genetic medicine space has had exceptional performance in terms of investment returns since 2015. And now more than ever, it's increasingly clear that manufacturing is not only critical to avoid industry growing pains, but absolutely essential to capitalize on multi-billion-dollar market opportunities. One of the themes as well from this conference is that scientific design and testing of products is super important for manufacturing. So we want to better understand the considerations that are heavily influenced by scientific choices. And for that, we now convene our panel, A Scientist's Perspective on Overcoming Manufacturing Obstacles. I'm joined by one of the key sources of value creation in the AAV-based gene therapy space, Dr. Brian Kaspar, who's co-founder and chief scientific officer for Modus Biosciences, scientific founder of Milo Biotechnology, co-founder of Celinex, and of course, scientific founder and chief scientific officer of Avexis. Thanks for joining, Brian.
Thanks so much, Gbola. Good to be on with you.
Great. So the format for this session is 40 minutes of fireside chat. If attendees have any questions during the session, feel free to type them in the question box at the bottom, and we'll try to ask on your behalf. So Brian, why don't we get straight to what a lot of us know you best for. You're the inventor behind Zolgensmo, the gene therapy for spinal muscular atrophy. I want to ask an intentionally broad question. So based on what you've seen so far at our summit, what insights do you believe you can bring to our manufacturing summit?
Yeah, well, thank you, Gbola, you and the Chardon team for having me on. It's a real privilege and an honor. And, you know, I think what I'd first like to say is that the conference has been wonderful. It's been great to hear so many of the companies talk about their paths forward. And you know what I think it's really exciting for me to share with you today is my perspective of how Avexis really built manufacturing and what my role was in it in helping to build the team to be able to crack the code on manufacturing. And of course, we'll be able to talk a lot about this in our fireside chat here. But really, it's kind of the scientific approach to think about the manufacturing, the biology that goes behind, the science that goes behind the work, preclinical work that leads into the clinical work. And one of the exciting things about Avexis was that we really didn't have silos. And we actually had all of the key components, call it manufacturing, clinical, regulatory, and science, all talking together and forging, the pathway forward. And that, I think, has allowed all of us to really get to see the challenges and all solve and crack the code to be able to figure out scalability and manufacturing and analytics. And, you know, I think one of the things that I had the privilege of really getting a front row seat was with the head of our regulatory, Dr. Jim L'Italien, in which we actually talked to the regulators around the world and to really think about the manufacturing processes and what the requirements were. So I think I had the privilege to basically be on the manufacturing floor, see the regulatory requirements, and help solve all of the problems.
Got it. So I think people know that Avexis took a different approach than others from building manufacturing, and in some ways established a number of industry standards in doing so. So can you explain a little bit around how Avexis accomplished this? You know, feel free to recap history because it's been some years now. We don't mind a history lesson.
Yes. Hold on one second. I think I'm having some technical. Let me just move here for a second, Gbola. Sorry about this.
No problem. Feel free to, if you need to turn off the video.
Give me just 1 second, Gbola. Sorry about this.
No problem. So due to the technical difficulties, I'll repeat the question. The inbound for Brian for us is hard to see. He's on audio just to save some bandwidth. So to repeat, I mentioned that Avexis took a different approach than others for building manufacturing. So Brian, can you talk a little bit around how Avexis accomplished that?
Yeah, and apologies here. We'll see if I can get my bandwidth expanded here to come back live. But happy to continue here, Gbola. I think we at Avexis had started the process at Nationwide Children's with a hyperstack methodology that got us into clinic and really utilized the expertise of the Nationwide Children's Hospital GMP facility to manufacture a very high quality virus that went in to the first patients of the clinical trial. And as we started to see clinical success, we realized at Avexis that we really needed to solve the commercial scale of this, realizing that hyperstacks really wasn't going to be the answer. And this is where the team really explored the vast abilities to think about scalability and really started teaming up. And I'll come back to this, taking down the silos and talk about how the manufacturing team really worked together directly with me to solve this challenge. Now, you had me with the AAV biology experience and making some of the world's largest amounts of AAV. And actually, at the time, being in hyperstacks, that was a feat. That was, to my knowledge, the first time hyperstacks had been, scaled for the manufacturing of AAV. So now we have this new challenge. We had to get out of hyperstacks. We had to figure out how to scale this. And a lot of the things that we thought from an academic perspective at the time were challenges. We teamed up with Andrew Knudin, who we had brought in to lead our manufacturing. And Andrew was just fantastic on the aspect that he had ran biological manufacturing for other companies. And really, that's where Andrew and I could understand each other's language of understanding the vast biology that goes behind AAV manufacturing, rescue, replication, and packaging, and some of the challenges that we thought we had, which were not really challenges when you talk to somebody who had been on manufacturing floors and thinking about 1,000 liters of media, something daunting for kind of an academic group, but really something that wasn't daunting to the manufacturing or the people who had ran commercial manufacturing. And that's where we put our heads together. And it was truly teamwork to solve the problems and think about scale, the upstream, think about the downstream processes, and also start building up analytics. I've likened this, Gbola. Here we had this fantastic clinical data. We were excited about this for patients. And here we had to crack the code to solve scale, to have downstream processes in place, and really have analytics. And a lot of this we were building from scratch. So I've always likened this as we were building the rocket ship as we were taking off. And this is really where the teams worked tremendously well together and really thought about this, not only from a scientific point of view, from a manufacturing and a commercial point of view, but also developing the analytics that we understood the product and really putting all of these pieces together with a regulatory framework of how are we going to show comparability and how are we going to have control over our processes. And I've said this before, so that batch 1 looks like batch 31, looks like batch 331.
Right. So, is it usual for a scientist to be involved in manufacturing when it comes to AAV, or were you taking a special role all along?
I think that was part of our magic, Gbola. No, typically the chief scientific officer, the scientist, is not going to be working hand in hand with the regulatory department or with the manufacturing department. At Avexis, we took down all of those walls and said, we're in this together. And I think that was our magic. Things that I would have never been able to be solved, Andrew's team were solving. When they ran into problems, they would pick up the phone call and say, we're having this problem. And most problems in this, and we'll talk later, Gbola, the manufacturing of AAV, and you've heard this from others, this is not a simple process. There's hundreds, if not 1,000 steps on the process of thinking about making the plasmids. How many mistakes can happen there? And they're scientific mistakes, to transfection, to the cell biology and the growth of the cells. I think something unique that we had was you had the scientist who had worked in AAV for decades, myself and others on my team, actually looking in the microscope, working with the manufacturing team to say, hey, we don't like how these cells are looking. Let's go back and try to mitigate what went wrong, where did it go wrong. It was not a punitive nature. It was really, how do we solve this problem? How do we get control of even splitting cells? And I think that in the health of the 293 cells is what we utilized. The health of these cells would be really important for the transfection efficiency. So there's so many places to make a mistake when you have 1,000 processes, or not be 100%. And you take cuts at every time that you make a mistake. And what Andrew, myself, and Jim and the team really did was to dissect each of those steps. And I think I brought the scientific perspective, because if you really think about it, there's cell biology going on, the health of the 293 cells, how one treats these cells, what is the proper density to be putting them into the bioreactor. When do we need to look at the cells? When do we need to change the media? All of these questions are really being driven by science. And that's where the teams worked really closely together. And I think this is how we pioneered the scale for Zolgensmo. And really then also, I don't think I can emphasize this enough, to develop the right analytical assays and knowing the right number of replicates to perform, so that we had the right number. And we hear this time and time again of people don't want to retest their product because they may get the wrong number. And Gbola, you want the right number, and you need to develop those analytical assays so that you get the right number on each and every batch of your product, and really understanding your product. Really can't emphasize that more.
Got it. So anything else to add on why manufacturing has been so difficult for AAV-based gene therapy companies?
I think this has been such a growing field, and so many places for errors or learnings to occur. We've heard this of we're learning on the fly throughout other presenters here. And I think that what we always try to do is put the lens on of trying to avoid the rabbit hole or avoid falling off the cliff. We were smart about this because we really teamed up with some first-class, top-class individuals within their field, but really thought about it from that scientific perspective to avoid. So I think, why has it been challenging? It's because the field has been moving so fast. There's so many new entrants into the field. And I think it's this secret mixture of, AAV biologists that really understand the viral replication process, that really understand the virus, to then combine that with people who know how to think about commercial manufacturing and try to avoid those problems. So I think this has been, we're early in the learning curve. People have taken different approaches. For example, Gbola, we were criticized because we didn't go out and hire the absolute AAV, expert to run manufacturing. But I think our approach really worked. At least it worked for us on the aspect of taking down the silos, working together as a team of scientists, of manufacturing experts, of analytical development experts, and quality to run these assays, all underneath the umbrella of expert regulatory guidance.
Got it. So let's move to opportunities. What excites you most about the gene therapy manufacturing space these days?
Yeah, thanks, Paula, for that question. I think first I'd start out is, how exciting that this space that many of us have worked in scientifically have seen the light at the end of the tunnel. And that is drugs can be produced, manufactured, and marketed in a commercial manner. And, I'll highlight just the brilliant success of Luxturna, of creating, vision to patients, and just a remarkable success that those patients have had. But then also to think about the success of Zolgensmo as well as CAR T products such as Kymriah, just really remarkable aspects towards patients. And when you think about just what excites me, Zolgensma, the first gene therapy to hit blockbuster status. Here's a drug in an orphan disease that just produced $920 million of revenue towards Novartis. That's a phenomenal success, in my opinion, for a gene therapy that's just come on the market. Maybe surpassing the financials here is, just how exciting it has been to see the thousands of lives impacted. Just really exciting that this can be done. And gene therapy is a drug modality that can have transformative, potentially one-time treatments towards patients. I think, to expand from there, because of those successes in the CAR T space, Luxturna, Zolgensma, and other drugs showing great promise, is just the amount of investment that has come into the space, which is opening up just tremendous opportunity, as well as the resources that are now coming in and the talents of CDMO partners, for example, that are bringing new technologies from end-to-end solutions. But when we think about how the field has just logarithmically expanded from thinking about 10 stacks to hyperstacks to now single-use bioreactors, the column chromatography, affinity columns that are now accessible today, really are exciting advances. Further, I think, just within the analytical space, for us to, understand the processes, but also the products that come out. Just for example, next-gen sequencing has really expanded, our abilities to understand the products that are produced. So tremendous opportunities, and I think tremendous opportunities for having huge impacts on patients' lives.
Understood. And so we're hearing a lot more recently about meeting scale for disease indications, including mass market indications. So Brian, do you feel that manufacturing is the last unsolved puzzle for gene therapy success in scale indications?
Yeah, Gbola, I think it's a great question. And I think a lot of people have said, this is the last unsolved puzzle. It certainly is a big piece of the puzzle to solve. But I think I'll bring maybe a slightly different perspective here on the aspect that, I think about this more holistically on the aspect of manufacturing is one part of success of a gene therapy product, but it's the science. Do we have the right target? Do we have the right serotype, for example, in AAV vectors? Do we have the right dose that's achievable for maximum tolerated or safe doses to go into patients? Do we have the right formulation and route of administration to properly target the organs that are being targeted for therapeutic success? So I think there's multiple prongs to be solved to really say, what is success in gene therapy? And I would say from a holistic point of view, each disease and each target really requires the right science, the right serotype in the case of AAVs, the right safety studies, the right promoters, the right vector design. There's so many parts that goes in that. Manufacturing is a large part, though. You need to understand your product, you need to be able to make it, and then you need to be able to get it out at scale to treat patients throughout the world.
Got it. So I want to talk a little bit about what a scientist can do to help ensure success. So you're currently involved in some new projects. So how are you going to do manufacturing better than others, and what differentiates your new endeavors?
Yeah, our thoughts based upon, going through these processes, seeing, where the bottlenecks are, where the problems are, how we view manufacturing is to lock down the process on day one. And, I think that the way our approach is to do our process development, our engineering, and have a process that we are happy about, to think that it can meet scale on day one. And I think the way that we also think about, gene therapy these days, after going through a biological license application, and I'm going to call it the daunting amount of information that is required to get across the finish line. I think that how we've taken that approach is to say, well, we know what the requirements are throughout the world on the analytics, on the manufacturing, on the PPQs, of manufacturing, and we work backwards, Gbola, so that we have those things in place, and that we're not trying to develop and build the rocket ship as we're just entering into the clinic. I'm going to try to get my video back on, just so that anyone watching, realizes that I lost a lot of hair on the aspect of going through these processes of, and I'll give just, one example, with a program that we had. People think about manufacturing, Gbola, as, you get the plasmid, you push the green button, and 3 weeks later you have a product that's in a vial and it's ready to go to the clinic. But let me just share with you, just one story of things don't always go the way that you think they're going to go. And, you're standing on the manufacturing floor and, you find out that you have a failure in one of your filters, or your pumps goes down, or you have a cracked bioreactor vessel. And, suddenly, you thought the process was supposed to be done at 3 p.m. or 4 p.m., and now it's 3 a.m. And you don't have your new shift coming on, or you can't move to the next, process step. The questions that starts coming up of, okay, what do we put this in? What to store the drug substance or drug product, and how do we store that until we can move it to the next step? Because it's 3 a.m., the team's already been working 15 hours, and you can't push anything more out. And that's where I think the science comes back into play, because to really understand, the virus, the aspects of what should we be doing to be able to continue that process and have ultimate success. So, they always say the sausage making is not pretty. And, I think as manufacturing groups continue to get experiences, they start finding out, where are the bottlenecks, trying to ensure that you never have that problem again, and being able to deal with this in a manner that, ensures the highest quality product, the safety of the product, and success of the product in patients. Those are the things that, we're going to take our real life experiences. And, some of these things, I think, Gbola, you don't see all of the problems and things that can go wrong on, 3 runs or 5 runs. You start seeing this when you've ran, 30 runs or 130 runs, and you start understanding, how to control those processes so that it is a seamless, manufacturing process that looks like pushing the green button and having, bona fide great virus at the end.
Understood. And so let's shift to regulatory a little bit, because manufacturing is so important in terms of where the FDA focuses before, advancing AAV-based gene therapy. So can you discuss the role of the scientist in optimizing that process? And then follow up and discuss whether you think regulatory standards are tougher now than in the past, whether it's 2015 or 2010. So Brian, you've been doing this for a while, so you can go back as far as you want on that question.
Yeah, Gbola, super, fortunate, maybe not so fortunate. I've got this, grow with this process of the science and the analytics and the regulatory requirement. And I also feel really fortunate to have worked with just such talented individuals who put their heart, soul, and lives into, developing Zolgensmo. I can't thank, those teams and individuals, hundreds of people who, just worked for one mission, and that was for patients. And it was to really do the right things and to develop this product. And regulatory aspects in all of the analytics are just critical and crucial towards thinking about these drugs getting across the finish line. I think, once again, how does the scientist help develop these? It's being part of the group to really think about how do we understand the product. We moved from Avexis, from qPCR, we made a collective decision, and I would say the absolute right decision, to move from qPCR, where we had very high variance, to droplet digital PCR, where we got really good control over our titers. And it goes in, how many samples would we run to ensure that we had confidence of the titers that were coming out. I think, just another really important aspect was the in vitro potency assay. That was developed in research and development. And, I give just a tremendous shout out to the team that we really tried multiple cell types, multiple approaches to be able to really think about how could we get an in vitro potency assay that was reliable and extremely useful for Zolgensmo. And the team did a fantastic job and developed an assay that's almost unheard of on the reliability and robustness of this assay. And I think this is where, once again, Avexis, part of the magic was taking down the walls of then, it's not just the scientists who hold on to something like an in vitro potency assay. It's working with the regulatory group and the quality group to hand that baton over, Gbola, to the team that is going to be executing, from a quality perspective, these assays and releasing drug product. And I think that was just, here we had now 2 teams that would never usually talk together, holding hands, learning and understanding these assays, and being able to pass the baton in a seamless fashion. I think just to to discuss, the thoughts of how the regulatory requirements have evolved. What I would say is that the regulators, FDA, EMA, around the globe, really worked hand in hand with us to develop these assays in the rigor that would be required to think about a biological license application. And I think, look, we were learning. The regulators were learning because we were, encroaching new ground. And I think that was, we took, and this is again with Jim L'Italien's leadership, we really took a proactive role to work closely with the regulators and be able to do the right things and control our products. So I think that, people have said, well, the regulators are getting more stringent. Actually, that's not necessary. They haven't changed the way in which they think. They've evolved, just as the field has been evolving on thinking about what are the right analytics and controls to put into place.
You mentioned analytics and controls. Can you talk about, again, what a scientist can do to innovate the process, the analytics? Ultimately that affects the product and cost. So what should a scientist do to learn from your example?
I'll just give, an example where I think the field has only started to think about it. And that is, how do we think about AAV manufacturing from the cells and the fidelity of the cells and how a scientist could be a part of that process of really understanding the rescue, replication, and packaging that occurs. And how can we do better? How can we have healthier cells, more transfectable cells, that ultimately gives the manufacturing team the biggest chance for success in increasing titers and increasing scale? There's so much science behind, Gbola, we could probably talk all day on this, of some of the, I think now people just take these plasmids and stick them into cells and, don't realize, just some of the molecular biology that was done by, Jude Samulski's team and Zhao to really develop these plasmids that enhance for the rescue, replication, and packaging, and ultimately the titers and the quality of virus that comes out. And I think there's a tremendous amount of work still to do and improve in this nature. So I think that those are just maybe a couple examples of how I see the scientist. And I always told my group, we are the group who probably knows the most about AAV. We know most about the AAV biology. We've put it into the most animals. We've developed assays that we really are now the service providers to be able to help the manufacturing team, to partner with the manufacturing and analytics, to really do things that pushes the field forward. And I think that's where we continue to learn. We continue to push the field forward on even analytics, thinking about how we can utilize mass spectrophotometry to better understand the characterization of our viruses, to now employ next generation sequencing to better understand the products that are coming out. That just leads to better clinical outcomes. So I think it again, there's the full circle of the universe, not just one single aspect of a company that works in isolation for success.
Understood. So let's talk about future innovations. And Brian, I think you've said that scientists tend to think about innovation continuously. I'm going to borrow that from now on. But what do you feel is the future of gene therapy manufacturing?
I think, as we're learning through this conference, as we're learning through our own experiences, is just the logarithmic growth that we've accomplished and continue to now refine and become better at the 1,000 steps. How can we automate some of these 1,000 steps to gain more controls in the manufacturing process? And then, I think, what is the future going to look like? Thinking about synthetic biology, this is things that we start to get excited about as scientists about, what is the future, platforms of manufacturing? We've cracked the code from, 10 stacks to hyper stacks to now bioreactors. And, we haven't stopped thinking from there. And we're trying to think bigger and bolder. And I see a bright future in this space. And you've got a lot of smart minds thinking about this. And, you can think about the cells. You can think about the platform of how we grow the cells and improvements that are going on there. We can think about the aspects of, downstream purification. I think, Gbola, maybe to wrap it all up, all of these advances have happened because of brilliant minds of science. Some of these capture columns first pioneered by Rob Cotton and group, who was doing this at the NIH as an academic, which are now we're seeing, very important towards, the commercial purification.
Got it. And what do you think is most underappreciated about the promise of AAV-based gene therapies?
I think that there's so much promise. What's underappreciated is the complexity here, and this is things that give me pause at the end of a day or at night of thinking about the speed that things are moving with the complexity of the processes. That I just want the field to move in the right direction, not make mistakes. We want to see more successes, and there's so many good people working on these challenges that we see today as a field, but really keeping focused on doing the right things, understanding our products as companies, and moving things forward in a safe and effective manner for patients.
Got it. And so we're running out of time, but a couple more. What important new trend might we be talking about at next year's Manufacturing Summit, very quickly?
I think that we're going to start seeing successes. The field, there's already so many great things going on within the field and encouraging clinical signs in some of these studies. I think that we're going to start feeling a little bit of the growing pains of this field, and having processes under control and ensuring continued success within this space. So I see, more advancements in the analytical aspects of the controls and real scalability, and the field really maturing. And we're starting to see that, even though we're still at early days, we're seeing a lot of newcomers. There's maturity that is building within this space. And I think you're going to see that in 2022.
Got it. And then the final question, and this is a non-manufacturing question, just something I've been curious about for years. So when did you first feel Zolgensmo was working? It was called AVXS 101 back then. But how many patients were actually treated before you said, this just might be working?
There was, hopefully my camera has come back on. People can see that there was a lot of praying going on, from day one of us entering the clinic. I kind of had the front row seat. I couldn't go to the clinic because of conflict of interest, of founding of Exis. Also, I'm not the clinician, I'm the science guy. But I had the opportunity to always watch Dr. Mundell, who's not only a friend, colleague, but really a pioneer within this field, who ran those first clinical studies of AVXS 101. And, I would always sit, I knew when the SMA clinic was, when patients would be evaluated. And I would always watch them come back. And usually, there was, no update because it was too early. And Jerry was always cautious, just because the kid moved their tiny finger, Jerry would say, don't get too excited. I've seen things go the opposite way. So always would sit there holding my breath. And I started getting encouraged because I started to see his clinical team start getting larger. Suddenly the pulmonologist, suddenly there's another neurologist, and another neurologist is joining the SMA rounds. And, I would say it was probably on patient, I'm going to say 6 or 7, where, we had seen the pinky move, but then we had seen a couple other things that we really encouraged. And I saw Jerry just walking back. He walked by my office and I saw everyone smiling. This was, 6 to 9 months into the clinical trial, 6 or 7 patients dosed, and people are smiling and laughing coming back from SMA clinic. That is not what clinicians used to do coming back from SMA clinics. This was a death sentence to parents and the families and the patients. So here they're coming back and Jerry and team are all smiling. And I'm like, About 5 minutes later, Jerry came back and showed me a picture, and it was of one of the kids in the clinical trial that was sitting up on the examination table. And that was the pinch me moment of, holy cow, everything that we, had thought about preclinically, non-clinically, thought that this would work. It was and prayed for it to work. This was the time where it's like, holy cow, this is doing exactly what we had planned for it to do. And it was then, Jerry's leadership on continuing the clinical trial, and also Avexis getting behind Jerry and team and really capturing the data in the manner, and I'll just give one example of starting to do recordings of the physical therapy and capturing patients doing these things that the natural history studies said that they shouldn't do. And I think it was at that moment that, I said, I have to go and be all in and, ensure that, we get this thing across the finish line. And, I just, maybe in my final few seconds, can't thank, the large number of people who were just integral, from Nationwide Children's to the Avexis team, Sean Nolan really putting together a fantastic team. And then the way that people just worked nonstop to ensure the success. I'm humbled and just super proud that, we as a team were able to pull this off. And, the fact is there's a large team that continues to pull this off day in and day out to get Zolgensmo out there to the world. And I really applaud this team.
Right. And I just want to say well done to you and your colleagues. There's really nothing better than saving a child from inevitable death.
Yes.
Thanks, Brian. We now conclude this session.